Let's do molecular docking of aspirin in 6-COX cyclooxygenase receptor. We have already downloaded 6-COX from PDB, and we have also copied the smiley string of aspirin in PubChem. So let's build the ligand first. Okay, so for that you need to go through tools, structure editing, and then build a structure. Okay, so here you need to select a smiley string. So I've already copied the smiley string of aspirin, so I'm going to paste it. Okay, so this is a smiley string of aspirin. You can rename the residue name as well. For example, here it is UNK. You can name according to the name of your ligand. So I'm leaving it default. Okay, no need to change. So make sure that you write a different you can say 3-letter code than that of we can say amino acid residue. So if it resembles, then the system will not detect your molecule. Okay, so better write a instead of writing ASP, you can write APS or something like that in case of aspirin. Okay, so once you do this, you just need to press apply, and then you will get the molecule in the background. So let's wait for the result. Okay, please wait, because of some kind of interruption it is not showing. So let me check it again. Okay, smiley string is perfect. Okay, let me apply it again. Okay, now you can see the aspirin on the background. Okay, so here it is the structure of 3D structure of aspirin. So now we need to minimize its you can say structure. The energy minimization needs to be performed. So for that you need to go to tools. You can go through you need to go to structure editing, and then you can move on to minimize the structure. Okay, so in this case you can just increase the number of steps to minimize it. So larger the number, the time will take the much greater time will be taken. So you just need to take care of this step as well. So for this workshop we are going to use 100 steps. So in your real research you can increase that to 1000 to 10,000 as well. Okay, so leave it default. Everything would be default in this case. You can change according to your you can say desire as well. So that's it, just press on to minimize. So next the hydrogen will be added to this molecule. So for that you just need to mark this, and then so here it appears protonation state. So as it is not a protein molecule, so we do not have any kind of amino acid residues, so better not to do anything, just leave it default. If in case of protein, you need to consider whatever you are doing in that particular protein. Okay, so for example in case of protein we will be talking about later actually. So let it be here now. Just press OK. So now the charges has been added. Not the charges is going to be added in this molecule actually. So in case of small molecules you can just press castigar, and the force field is applied over there. The amber force field is there, so you just need to press it. You need to click on to castigar charges and leave it default, and then press OK. So next is the net charge of this molecule has been calculated by Chimera. Okay, you can just press OK, and then you can see the structure would be a little bit twisted at the end to minimize the structure. Okay, look, it is the minimized structure of this particular aspirin molecule. So you need to save this molecule in mol2 format. So I've already allocated one folder here, so I'm going to save it as a prepared aspirin mol2 file. So you can do it, and then you just need to save it. Okay, so finally it has been saved onto the particular folder. Now let's prepare protein in the similar fashion. So let's close the session first. Let's go to file, let's open protein, that is 6-COX. okay, here it is. So it's a 6-COX molecule. So we have already got the information of 6UX in the PDB, protein data bank. So it says it has two chain and both the chain are similar in nature. That means it is a homodimer. So you can use one of the chain. So I'm going to use A chain. So let's delete the B chain, the bonds, and delete. So here is the molecule. Okay, so I've told you about the parent ligand present in this COX receptor. So there are three parent ligand NAZ, HEM, and S58. So actually in the experimental data we have already found that S58 acts as an inhibitor in case of cyclooxygenase in this particular cyclooxygenase. So let's focus on that inhibitor, and you can say let's screen aspirin in place of that S58. So let me delete other residues. For example, heme. Heme has been deleted. Now let's go to NAZ as well. There are three NAZ in this chain, so let's delete it as well. So now you can see just S58 only. Okay, so now we are going to use S58 as a parent ligand, and instead of S58 we are going to dock aspirin molecule. Okay, so for that let's prepare this protein first, energy minimization and everything. So for that you need to go through surface binding analysis and then go to dock prep. Okay, now you can see 6UX has been selected over there. No any non-standard residues, or you can say foreign residues, such as solvents and other things needs to be deleted. So here it has been selected as well. So let it be placed over here, and you can see incomplete side chains. So side chains needs to be deleted in some aspect. If you know the detail about the side chain in particular protein, then you can go through this. If you do not know, then just leave it default. Okay, so next is addition of hydrogen, addition of charges, and you can save that into mol2. I'm not going to save this into mol2, so I'm on marking it. Later on I will save it into PDB format. Okay, in case of ligand you can save into mol2 and then protein into PDB, or you can make it save in both the proteins and ligand can be saved in similar format, for example mol2 or PDB. Okay, better you can save on PDB for protein and mol2 for ligand molecule. Okay, so that's how we can do it. So press OK, then after the hydrogen would be added in this step. Okay, so just press it default, you can make it default. Hydrogen would be added into the molecule. So here you can see the protonation states that I've already told you in the ligand as well. So in case of protein, it is very important. If you know, if you are working on the particular residue in the protein, then you can go through the protonation state as well. For example, here it is mentioning histidine as a protonation state residues, but you can change it. Okay, so if you know it, or else leave it default, and then leave it default, just go to press OK. So now you can see we are going to add a charge as well. Here you can see the Ember force field is applied and gastric charge are marked. Okay, so let it be here. And now S5, the net charge of S5 has been calculated over here. So press it OK. Now it has been well prepared. You can see the result. It's running there, and once it is completed it will be shown. Okay, now it's the protein has been well prepared. Now you can save it onto PDB, and then you can save it onto you can say you can save as a prepared protein. You can later use this. Okay, let me save prepared protein. Okay, let me save it. So in the same tab, now let's open ligand molecule as well. Okay, prepared aspirin. Okay, here you can see aspirin has been integrated into this workplace. So let's select. Okay, let's select S5 so that we can prepare a grid box over there. Okay, it's S5, and let's prepare a grid box in this particular region. Okay, so let's, for that we need to go through. Okay, let's go to tools and then surface binding analysis and then AutoDock Vina. So you can see it right now, right there. So click on to browse. You need to give a output file location as well. The PDBQT file will be generated, so you can name it like aspirin docked in COX. Okay, so let it okay, now it has been given there. Okay, now we have already uploaded our aspirin as well. We need to change the ligand notation as a small string over there. And now let's see, let's resize, let's create a grid box. Let's say make it okay 23. You can write a random number. Okay, I'm writing okay 24 44. Okay, and then let's see 22 and then 24. Okay, now you can see a grid box has been generated. I have used this I have used the dimension of the box as I've already performed it before, so I've used it in the standard way, but you can easily change it. Okay, you can remove the you can drag this box using the, you can say, using the wheel of your mouse. Okay, in this way you can just you just need to tap on the wheel and then you can drag it, the box will be moved. Okay, and then to increase the size and everything, you can just go through the wheel and then just click onto the wheel and then drag it. Okay, the size will be increase or decrease. Next, you can also increase or decrease the size. You can also increase or decrease the size. You can also increase or decrease the size. For example, let's make it bigger and then allocate it further. Okay, okay, it's a grid box. Okay, I've changed the dimension of grid box. So if I need to move this grid box to the active site region, so I can do this. Okay, so you just need to prepare a grid box in such a way that this molecules is under the inside the box, and make sure the box is smaller in size. Okay, just to cover the active site residues present in the bucket. Okay, make it as smaller as possible. So here it has been created perfectly. So you can see the aspirin molecule is inside the grid box, and then all of the active site residues are also present. So that is how we can do it. Okay, so that's it. Okay, let's check the dimension and everything. So dimension is, here it is, 21, 24 and 49. Okay, and then next is 22, 22 and 20, 28, 22 and 26. So let me change into let's say 23. Okay, make it 24. Okay, make it 44. Okay, 22. Okay, make it 22 as well. Okay, 22 and then 24. Let's say 24. Okay, so you can see it has been perfectly placed inside the grid box, and some of the residues, most of the residues are also been inside it. Okay, so here it is. Now we can easily perform molecular docking. You can run the AutoDock Vina as well now to perform the molecular docking to get the binding energy and everything. Okay, so here you can okay, let me see. Okay, now you need to copy this dimension. Okay, send the dimension of center and size as well, size of box as well, so later you can use it. Okay, you can modify the numbers based on the result obtained after molecular docking. Okay, so here it is. Now next is receptor option. You just need to make it true or false. based on this default values. You can also change that into true as well. In this is step, ignore non-standard residues, so you can make it true as well, because we are not dealing with any kind of non-standard residues. All of the options are correct. Add hydrogen in chimera, so it's true. If hydrogen has not been added before, then in this step as well you can easily add more charges and remove non-polar hydrogen, more charges and remove lone pairs, ignore water. So so we do not do molecular docking inside the water molecule, so we can just we just need to ignore this water. Ignore chain of non-standard residues. I've already told you about the residues as well. So next is ligand option, so you can see more charges and remove non-polar hydrogen, true. More charges and remove the lone pair. Okay, in this aspect you can go through the options. Okay, so next is advanced option. You can see number of binding modes. So after performing molecular docking, you will be getting 9 different docking poses given by AutoDock Vina here in this case. And next is exhaustiveness of the source. Exhaustiveness means the reliability of this result, so you can mark it to 8. You can change the data, and maximum is the 8, so let's make it to 8 as well. And next is minimum or maximum energy differences, so you can just make it to 3, or you can just increase whatever you want. Okay, so and the default mode, it is selected C, so I'm not going to change it. Okay, next is, so we need to give the pathway of the Vina exe, that is when already downloaded. I've already given you the link as well, so you can just go through the browse and then go through the folder where your Vina exe file is located. Okay, once you do this, you just need to okay, in this I have forgot to remove this parent ligand, so let me go through residue. So delete this, it hasn't already selected, so let's delete it. So here is it. Now you can run the AutoDock Vina. Okay, so or okay or apply. Okay, do not press okay and apply for a couple of times, or just press one of them, okay or apply. Okay, okay, I pressed okay, so let the Vina be running. Okay, now you can see the result has already been obtained. So you can see the binding energy of different conformation. There are 9 different conformations that has been generated, and the binding energy has been resulted to be minus 6.9 kilocalorie per mole, and so on. Okay, and here you can see the RMST lower and upper value as well. So in case of RMST, so RMST means root mean square deviation. So original value, original conformation is this one, the first one, and based on the, in comparison to the first one, the different conformation are generated, and you can just go through this. Okay, and you can check each and every conformations. Okay, and the RMST value, you can just look through it. So here you need to be clear about the RMST value as well. So RMST means root mean square deviation, so the value needs to be lesser than 2 angstrom for a perfect conformation. Okay, if it is away from the original pose, then it doesn't work in that sense. Okay, because it can have on a specific binding, and a lot of other on specific sites would be incorporated into the interaction, so that is not feasible in nature. So there needs to be some kind of reliable interaction in the active site regions, so the RMST value of less than 2 is more favored. Okay, so you can see the first conformation is okay, the second one is also fine, but the upper value is quite greater than 2. So okay, this one is perfect. Okay, the third 1 2 3 4 fourth one is fine. Okay, and so on, you can screen it. Okay, the fourth one and first one looks better. Okay, let's check this first conformation. Let's check the hydrogen bond of this molecule. So you need to select the intramodal. You can change the hydrogen bond coloration as well. Okay, based on what color you like. and you can label the hydrogen bond with distance as well. So here I need to make you clear about the hydrogen bond distance. Okay, so for biologically relevant, or you can say biologically significant interaction, the hydrogen bond distance needs to be lesser than you can say 3 angstrom, or better than better it would be lesser than 3 actually, but in many of the literature they have written 4 as well. So better you can see, better you can analyze the lesser than 3 angstrom hydrogen bond distance would be a better option. Okay, so you need to check for the hydrogen bond length as well if you are looking for biologically active complex molecule. Okay, so you just need to select the include intra-residue hydrogen bond as well, and now everything looks fine. You can just press onto OK. Okay, maybe running. Okay, fine. Okay, you can see in case of this conformation, okay, this conformation there are hydrogen bonds, but not in the first one. So you can go through the fourth conformation. It is found in the RMST as well. Okay, lesser than 2 angstrom. So there are 2 different hydrogen bond present over here. Okay, so you can check this, the length of hydrogen bond and everything. Okay, actually it's one only, it's 2.073 angstrom. Okay, so it looks perfect as well. If you want to check other conformation, then you can just click here. Okay, there is no hydrogen bond in the first one. Second one, it has it doesn't have, okay, it doesn't have any hydrogen bond as well. The third one has hydrogen bond. Okay, let's see the fourth one. We have already labeled it. Fifth one, do pauses. Okay, so now let's screen the fourth confirmation. Okay, that's it. This we are focusing on. So now let's model this confirmation. Okay, so aspirin had already been selected, so let's label it. Okay, let's okay, it has not been selected yet, so you can press on to click. Okay, and then you can press into upward arrow option. So it has been selected, aspirin has been already selected. Just you can go through select, and then you can create a zone of lesser than 5 angstrom. Okay, the zone is selected based on the favorable region. Okay, so for less than 5 angstrom region is more favorable for any kind of biological interaction, so that's why we select it. So okay, so it's a most favorable region in this case. Okay, now let's go to, okay, let's export some side chain molecule as well, side chain bases. Okay, so okay, these are the side chain molecules. Okay, it has a 2 hydrogen bonds, sorry. Okay, later on we will label it, don't worry. Okay, let's label the molecule as well. Okay, label it residues. Okay, name and specifier. Okay, you can say it. Let's change the color of the label as well. Okay, color, all option. Okay, residue label. Okay, let's give it red coloration, yellow, green. Okay, make it red. Okay, fine. Now you can see where it has been. You can see 2 hydrogen bond present over here. Maybe the valine. Okay, valine 250. Okay, 523 in A chain, and the hydrogen bond distance is 2.073 angstrom. It looks fine. And next, okay, let me check it. Okay, here it is. The hydrogen bond distance is 2.080, and it has been linked with serine 530. Okay, so there are 2 hydrogen bond for this particular conformation. Okay, in this way you can go through. the labeling, and you can change the color as well. Okay, let's change the color of entire. Okay, you can see it. You can change the color of label only. Now let's go to color and all option, residue label. Okay, what would be the best one? Magenta. Okay, magenta. And let's change the size of the label. Okay, fine. So you can see it has been well labeled as in bone distance. So you can also change the color of as in bone distance and everything. So let's change the color of aspirin as well to make it look more. Okay, to provide a better contrast, let's change the color. Okay, go to color and then make it red. Okay, so you can see both the hydrogen bond over here 2.080 and 2.07, so it is perfect result. Now for the validation of your docking protocols, so in case of this particular molecule, it's a standard molecule and it has parent ligand embedded into it, so you can easily to validate this, you can easily redock that parent ligand. For example, in case of this molecule, S5 is a parent ligand. You just need to go through the you just need to go to PopChem and find the name of that S5 molecule. Okay, from PDB as well. From PDB you can get the name, and then you can go to you can say PopChem to find the smiley string and come back and then prepare whatever we did before. Prepare that ligand molecule and then redock into the active site region. Okay, and then once you redock, you will get a confirmation of that parent ligand as well, best confirmation, where hydrogen bond will be there, because hydrogen bond is the most required bond for any kind of biological interaction. And it also depends upon what kind of ligand molecule you are using and what is the receptor as well, because in some cases hydrophobic interaction are also valid. Okay, so it depends upon what type of protein you are working on. So in this way you can redock that, and then after performing a redock, after redocking that molecule, you will get one best confirmation, and with that best confirmation and the confirmation present in the database, you just need to superimpose both the molecules and then find the RMST value lesser than 2 angstrom. Once you find the RMST value less than 2 angstrom, you will get the perfect result. Okay, that means your docking protocol will be validated. And next step about validation of your docking protocol is to use okay, you can use positive control as well as negative control and do the molecular docking thing, and then you will be getting binding energy and everything there, interaction pattern. Just look for two things: binding energy and interaction pattern. In case of positive control, the binding energy will be the laser binding energy will be present over there, and in case of minimum binding energy actually. And next, in case of positive control, in negative control, you will get a different result. Okay, and the interaction pattern would be different from positive to negative control. In case of positive control, it will be linked onto the key residues present in the pocket, while in case of negative control, it can have some kind of unspecific binding. So in this way you can validate your docking protocols. Okay, and there are other methods as well in case of validation, but I follow these two methods and I highly recommend them because it gives a reliable result. And I perform molecular dynamic simulation after doing this kind of you can say validation procedures, and so I got the best result in the molecular dynamic simulation. So you can also follow the similar pattern for validation thing. Okay, so this is it for the entire workshop. So if you have any questions, you can even save this molecule. Okay, save this entire complex and everything. So if you have any other question, then we will be moving on to Q&A session now. Thank you. So now let's deal with some of the questions that has been asked by Mr. Jamal. So he has asked that if no NAS structure of protein is present in the database, then how can we analyze how can you build the model and everything. So in that case, we can use homology modeling. You can develop your own model, okay, comparing to the standard protein present in the database, and then you just need to superimpose that the standard structure or the standard structure of protein with your model. And then if everything is fine over there, if the RMS value is lesser than 2 angstrom, even lesser than 0.5, that would be the best one, okay. And then you can also see the dimension, you can also see the dynamics of your chain and everything by using molecular dynamics simulation. Or else if you do not have that facility even, then you can just superimpose two of the protein molecule, okay, your model and a standard model. And if you get the desired RMST value lesser than 0.5 or even closer to 0, then that would be the best one, okay. So in that way you can easily develop your model and then further analyze that result on, okay. And next is he was also concerned about you can see uh, how the structure, okay, how we can predict the active site residues of non-standard protein or non-standard or non not fully solved structure in the database. So in that kind of protein molecule, you can get a sequence from the database. For example, in case in from the NCVI or even from any other databases, okay, you just need to get the sequences and multiple sequences from different organism of the same protein, and then you can do multiple sequence alignment, and then you just need to see the conserved region. So the longest conserved region would be the active site, and in that case you can just mark the longest conserved region. For example, you can just mark the amino acid chain, amino acid residues, okay, from this to that, and then you can just mark it, and you can just come on to the visualization tool, and then you can just label those amino acid, and you can easily see the you can see active site residues and active site pocket as well. You can also create a pocket using visualization tools such as Chimera, or you can say Chimera, or any other tools, Discovery Studio Visualizer, okay. So in this way you can easily screen that, okay. And he has also asked that if ligand is bound to somewhere else, then what would be the option. So if you follow the exact step, okay, if you follow the proper step, then you would not find any unspecific binding. Or in case of this kind of result, okay, if you find any unspecific binding nature of your ligand molecule in the pocket, then that is not a valid result. So you just need to trace back your protein as well as your model, and then you just need to superimpose again, and then look for the you can say Ramachandran plot as well. Look for the favored region if it is quite better, okay, if all of the residues fall under the favored region or not. Maybe more than you need to look for more than 90% of favored region, and you also need to look for the mole probability score and all that thing, okay. Once it is validated, then only your result will be valid result, okay. Everything is sorted in that way. So